This is part one of the lecture on important biological macromolecules. This is chapter 3. All right, there we go. So this is another chemistry chapter, but we're focusing on macromolecules. And there are 4 macromolecules, carbohydrates, proteins, nucleic acids, and lipids. And since I don't see a slide for that, I guess let me write it down here. So the 4 classes of macromolecules are carbohydrates, lipids, proteins, and nucleic acids. So in this chapter, we'll go through each of the 4 classes of macromolecules in detail. We're beginning with proteins. Proteins are one of the most abundant organic molecules in living systems and have the most diverse range of functions. And proteins are polymers of amino acids. So we would say the amino acids are the building blocks or the monomers, and they're joined together. Sorry, hold on. Okay, the amino acids are the monomers, which is another word for building blocks, and they're joined together in a chain to form the protein. So we call the protein a polymer. A polymer is a chain of monomers. So for proteins, protein is the polymer and amino acids are the monomers. So if the amino acids are the building blocks of proteins, then we need to also learn about the structure of an amino acid. And here we have amino acids are the monomers that make up proteins. So each amino acid has a central carbon, and we know that carbon can make 4 chemical bonds. So the carbon bonds with a hydrogen, and then it bonds with an amino group, which is a nitrogen and 2 hydrogen atoms, and then a carboxyl group, which is an acid. That's what makes it the amino group makes it an amino, and then the carboxyl group makes it an acid. And then every amino acid, there are 20 different amino acids. Every amino acid has an R group, which is a variable group. So R can be any element or any group of chemicals. It's variable. Each of the 20 amino acids has a different R group. Okay, the bond that links amino acids is called a peptide bond, and amino acids are linked together through a synthesis reaction that we know as a dehydration reaction, because it removes water during the reaction. Water is a product, in other words. So the reactants are the 2 amino acids, and then the product would be water and the peptide. In this case, this is a dipeptide because it only has 2 amino acids, but a polypeptide would have many amino acids. And the only difference in a peptide and a protein is the peptide is just a chain of amino acids, and the protein is a chain of amino acids that has a complex shape. So we also need to talk about protein structure because the shape is critical to its function. There are 4 levels of protein structure. Some proteins only go up to the 3rd level, but some actually have a 4th level. So the levels of protein structure are primary, secondary, tertiary, and quaternary. So the primary structure is simply the sequence of amino acids. Now, you're just looking at the abbreviations for each amino acid, but like this is glycine, this is isoleucine, valine. I think this one is glutamate. I'm not sure. There's several that start with GLU, but cysteine, alanine, serine. These are all different amino acids. So when you just look at the sequence of amino acids that forms a chain, that is the primary structure of the protein. And the sequence of amino acids is determined by DNA. There are genes. The genes code for particular amino acids. The secondary structure of the protein is that chain that we just looked at is either formed into a helix or a pleated sheet, an alpha helix, which is a helical structure, or a beta pleated sheet. You may have made a paper fan in your life and you've had to fold the paper, so it's pleated like that. And the alpha helix is just a spiral shape, kind of like DNA, but it's not two strands, it's just a single strand. So this is the secondary structure of the protein. And the tertiary structure is the three-dimensional structure, the final three-dimensional structure of the protein. So if you look at this red line right here, the wavy line that you see, that can either be the alpha helix or the beta pleated sheet arranged into that particular three-dimensional structure. I always explain it to students like this. If you remember that there are phones with cords on them, so this example was better years ago when we had phones that were not cordless, but phones can have cords. I'm sure you've seen a cord on a phone or on a hair dryer or something. Well, they might not be the way a phone cord is, but a phone cord, I've got one right here, looks like this. Look up at where I am. There we go. This is what a phone cord looks like. Okay, I'm not really good with this. So this is a phone cord. So that would be like the alpha helix structure, the secondary structure called the alpha helix structure. And if you took that phone cord and tied it in a bunch of knots, that would be represent the tertiary structure. Okay, the quaternary structure is only found in some proteins, but that is when you take two tertiary structures and link them together. So they're formed from several polypeptides. One example of a protein with a quaternary structure is the protein hemoglobin in your red blood cells. Hemoglobin carries oxygen, and hemoglobin has 4 polypeptides that are linked together. So that would mean hemoglobin has a quaternary structure. And this is a good picture. It shows you the primary structure is just the chain of amino acids. The secondary structure can be a helix or pleated sheet. The tertiary, you take that alpha helix or that pleated sheet and you kind of rearrange it into a three-dimensional shape. And quaternary, you take 2 or more tertiary structures and link them together. Proteins can be denatured by heating or by change in pH. Change in temperature, change in pH, or exposure to chemicals can denature a protein. And when the protein is denatured, it loses its shape. And if it loses its shape, then it won't function like it's supposed to. So it still keeps its same amino acid sequence, but it loses its shape. And denaturation can be reversible or irreversible depending on the situation. And denaturation does lead to loss of function. When the protein is denatured or the shape is changed, it cannot function. Protein folding is critical to its function. So there are helpers known as chaperones that associate with the protein during the folding process. And then they disassociate or disattach, I guess, from the protein once the folding has completed. So a chaperone is something that helps a protein to fold correctly. And this chart gives you a list of many different protein types and their functions, examples. So all enzymes are proteins that speed up chemical reactions. Some examples here are amylase, lipase, pepsin, trypsin. Pepsin and trypsin help to digest proteins. Pepsin is particularly found in the stomach. Trypsin is found in the small intestine. Amylase and lipase help to digest starch and fat. So these are enzymes that help digest, but there are enzymes that catalyze many, many chemical reactions. Every chemical reaction in the body has an enzyme that helps to catalyze it. There are some proteins that transport. Hemoglobin is an example of that. It transports oxygen throughout the blood. There are structural proteins like keratin found in our skin. There are hormones that are proteins. Now, all hormones are not proteins. Steroid hormones are lipids, but some hormones like insulin are protein hormones. Then there are proteins used in defense. Immunoglobulins are also called antibodies. That's another word for antibodies. Which function in our immunity and our immune system. And proteins can also be contractile, help our muscles contract, actin and myosin, and they can be storage proteins like egg white, which is also called albumin. Now we're on to the second class of macromolecules called lipids. Lipids are nonpolar covalent molecules, meaning they will not dissolve in water. And they're hydrophobic, which means water-fearing. And types of lipids include fats and oils, waxes, phospholipids that are found in our cell membranes, and steroid hormones. So a fat is a stored form of energy, also known as triglyceride. They're made up of fatty acids, and fatty acids look like this. They're a hydrocarbon chain with a carboxylic acid group attached. And the fatty acid may be saturated or unsaturated. If it is saturated, it only has single bonds between the carbon atoms in the hydrocarbon chain. Single bonds, meaning that those carbons are only sharing 2 electrons or a pair of electrons. But if it were unsaturated, at least one of the bonds between carbon atoms would be double. So if this bond was double, you would take away this hydrogen, and you would take away this hydrogen. Sorry, I'm trying to mark them out. But anyway, if you had a double bond between carbons anywhere in the hydrocarbon chain, you would call it unsaturated. The fatty acids combine with glycerol, and that's what forms the triglyceride. And here is a much better picture of unsaturated and saturated fatty acids. This is a saturated fatty acid called stearic acid, and you can see, if I move my face, you can see that, you can see that there are only single bonds between carbon atoms in the hydrocarbon chain. But if you look at unsaturated fatty acids, here is a double bond between carbon atoms in the hydrocarbon chain. And that also results in less hydrogen atoms because the double bond removes 2 hydrogens from the chain. And it results in folding. It results in the chain being kind of bendy or foldy, and that makes it tend to be a liquid. Unsaturated fatty acids are usually your oils, and saturated fatty acids are usually your solid fats like butter. Trans fats are man-made, like if you don't get natural peanut butter, if you just get regular peanut butter, that uses a trans fat, and so does margarine. Margarine is a trans fat. A trans fat is an unsaturated fatty acid that has been straightened out by man. So it will be solid at room temperature, even though unsaturated fatty acids are normally liquid at room temperature. A trans fatty acid will be solid at room temperature. And then this is an omega-3 fatty acid. Just so you can see it.